In 1859, Charles Darwin introduced the concept of evolution as the survival of the fittest. More than 160 years later, I applied this same principle not to animals or plants but to microorganisms. Specifically, I worked with Clostridium acetobutylicum, a tiny bacterium capable of producing biobutanol, a renewable fuel with the potential to replace petrol. But there's one problem: it struggles to survive in its own toxic product. Today we would like to present on our innovation, which is next-generation microbial engineering, activation-enhanced ALE for high-performance biobutanol production. To understand the fundamentals of my research on biobutanol production, we must first look at the global energy demand. To date, the world has produced a total of 153 billion liters of biofuels, which include bioethanol, biodiesel, and biobutanol. Among these three types, biobutanol is considered an advanced biofuel because it has a lower autoignition temperature, is less corrosive, evaporates more slowly, releases more energy per unit mass, and can blend seamlessly with conventional fuel without phase separation. Generally, biobutanol is produced through biological process called ABE fermentation with the aid of microbial species Clostridium acetobutylicum. However, despite biobutanol having superior properties, it is still not economically feasible due to several constraints. Therefore, this brings to my problem statement, whereby the main problem rise from biobutanol production is its toxicity. For instance, at high concentration of butanol it causes inhibition towards the Clostridium species, which leads to denaturation. Most researchers now are focusing on enhancing the strain to tolerate the butanol concentration. However, the conventional method that focus mainly on metabolic engineering requires expertise, and in order to enhance the strain, full understanding is required down to metabolic gene level, which is quite troublesome. Therefore, our solutions are more derived towards engineering based. We as engineers, we want something that is cost effective, easy to handle and provides better productivity, right? All of this can be achieved through ALE strategy. So, what is ALE? ALE is actually a technique of improving the strain through serial cultivation with different selection pressure until it can be adapted to end goals we want. For instance, in our study we emphasized on improving the strain tolerance level by subjecting to high biobutanol concentration that starts from 15 gram per liter and end at 23 gram per liter, that will be further elaborated in the upcoming section. Now, let me introduce the main hero of my research. It is our locally isolated strain from Salanger, a biological material that I aim to develop and enhance, known as Clostridium acetobutylicum YM1. The term YM1 reflects a personal story. It stands for Yemen, the origin country of the researcher, while Malaysia is where the strain was successfully isolated. One of the unique traits of this strain is its ability to adapt to both aerobic and anaerobic conditions. It also has a proven record of utilizing various types of agricultural waste, making it highly versatile for sustainable biofuel production. To further support the necessity of conducting this research, these are some of the examples from previous studies that portrays the constraint caused by butanol toxicity, in which the maximum inhibitory concentration of most of the Clostridium species are below 15 gram per liter. These are some of the achievements of applying ALE method from previous studies. Notice how very limited study related to strain improvement of Clostridium species in biobutanol production is reported using ALE, which therefore further motivate our research to incorporate ALE in mitigating biobutanol toxicity. This research covers three main objectives, whereby the first objective is to evaluate the success of conducting ALE strategy, the second objective is on strain validation through multiple validation strategies and also transcriptomic analysis, and the third objective is to evaluate the scaling up strategy in 2-liter bioreactor. In research methodology, initially the TYA medium and bacteria were prepared, then maximum inhibitory concentration of our strain was determined, which is vital to know as the starting point of ALE. We start the adaptation with 15 gram per liter because we found that this is the inhibitory butanol concentration for our strain. In ALE strategy, once the strain has acquired the strength to sustain at that particular concentration, then we move up to next concentration. This is continued until the strain reached its limit and was proceeded up until 23 gram per liter of butanol. So, how was the adaptive laboratory evolution strategy executed? The process began by adding 10 milliliters of inoculum into TYA medium. Based on the working volume of the medium, a specific amount of butanol was added according to the desired concentration. For instance, for 17 gram per liter of butanol concentration, 2.11 milliliters of butanol was added for 100 milliliters TYA medium. The samples were then monitored over several days by measuring optical density using a UV spectrophotometer. Once the OD value reached above 0.7, the culture was transferred to a higher butanol concentration, in this case 18 gram per liter of butanol concentration. This stepwise adaptation continued, with the strain being exposed to progressively higher concentrations, until it finally reached its inhibitory level. Okay, after I applied ALE strategy, we can see improvement in the adapted strain compared to the wild strain, in which for the biobutanol production, the butanol produced in the adapted strain was higher compared to the wild strain. However, it is apparent that the yield and productivity for 17 gram per liter is quite low. But I still feel like the strain is capable more than this concentration, and maybe we could do something in the method, maybe some sort of modification. So therefore, I came up with a new proposed technique that can enhance cereal cultivation method during ALE, which is called activation approach. This method was filed for patent protection and currently undergoing substantive examination, SE, and also has been registered for copyright. The basis of this approach is that instead of conventionally transfer the strain directly to the subsequent concentration, the strain is actually subjected to TYA medium prior to next cultivation. One of the necessities of conducting this approach is to prevent overexhaustion of the strain and also to promote longevity of the strain to tolerate higher butanol concentration. Then, after I applied my proposed novel method, we can see significant improvement, and the strain can adapt up to 22 gram per liter of butanol and manage to produce 11.51 gram per liter of biobutanol. This best adapted strain of 22 gram per liter was further selected for validation and scaling up strategies. The 22 gram per liter, which is the best adapted strain, was then validated through multiple validation strategies cover in terms of best growth, morphology, and also fermentation performance. In terms growth assessment validation, the adapted strain of 22 gram per liter able to produce almost same amount of biobutanol as during ALE strategy, that further proves the viability and stability of the adapted strain of 22 gram per liter. As for fermentation performance, in this part one want to compare the viability of the adapted strain with and without butanol addition. Based on the result, we can see the impact of butanol addition in restricting overall production yield, and also the strain without butanol addition shows superior butanol production, productivity, and yield compared to the strain with butanol addition. To further understand the differences, here we can see that even when butanol was added to the medium, the adapted strain can produce high butanol without impacting the viable cell. In this case, butanol was not added to the medium, and the butanol produced was quite high for a lab scale result, which was up to 21.26 gram per liter of butanol produced. Talking about viable cell, now we move on to determining the viable cells of the adapted and wild strain using hemocytometer. So, in this case we added butanol, and we can see here in adapted strain despite butanol was added, the viable cell is very high compared to wild strain, which clearly shows sign of inhibition. Then we did comparison test with hygiene monitoring, which a new fast method to count cell, in which it only requires us to insert then sample and go through several steps, and we can the full cell counting. Based on the result obtained, the CFU per ml for hygiene was slightly higher but still within the range of the hemocytometer method. Moving on to molecular and structural characterization, SEM-EDX was executed, and from here we can clearly see in SEM the adapted strain is much smaller and has rough surface due to butanol stress, which is same as previous studies. As for EDX, microelements that helps in adaptive mechanism in adapted strain is seen to be visible, such as calcium, magnesium and zinc. Another type of characterization, functional group characterization, FTR, was executed, and several strong peaks is visible and shows distinct difference compared to wild strain. For instance, amide bond at wavelength 1650 nanometers shows enhanced protein expression due to stress-induced enzyme production. Apart from that, this transcriptomic analysis confirms that our adapted strain is not only phenotypically improved, but also genetically reprogrammed for enhanced performance. We observed significant upregulation of key genes such as ADHE, which drives butanol formation, and CRT, which is essential in the butanol biosynthesis pathway. This molecular evidence clearly explains the strain's ability to achieve higher tolerance and productivity beyond conventional limits. On the other hand, Scaling up strategy in 2-liter bioreactor revealed high amount of butanol produced that further highlights its feasibility for industrial application for biobutanol production. This study successfully demonstrates that our activation-enhanced ALE strategy enabled the strain to overcome its natural limitation, achieving up to 22 gram per liter butanol tolerance with significantly improved performance. The robustness of the adapted strain was further validated through multiple approaches, including transcriptomic analysis, confirming enhanced tolerance at both physiological and molecular levels. Most importantly, this improvement translated to real-world potential, where scale-up in a 2-liter bioreactor achieved up to 29.41 gram per liter, proving its strong industrial applicability. As for the economic assessment and invention advantages, our study has managed to enhance biobutanol production with significant improvement from lab scale to scaling up, and also butanol tolerance in which there is 7 increment, which is by far the highest compared to previous study that only has 1 to 2 increment improvement. In terms of cost estimation, it is also proven how cost-effective employing ALE strategy in contrast to genetic-based strategy. In terms of novelty, our adapted strain, the first Clostridium species in the world that could tolerate butanol concentration up to 22 gram per liter. We also have our own method produced and currently going through patenting process under my IPO. Not only that, in terms of commercialization potential, we currently undergoing 2 collaboration, one from Industry Free the Seed where they want us to utilize rice straw pulp for both bioethanol and biobutanol production, and also one international collaboration with Papua New Guinea University in which they want us to utilize copra meal for biohydrogen and biobutanol production. These further highlights the commercial use of our adapted strain in industrial setting. This technology has already achieved TRL 5 to 6, demonstrating successful performance in relevant and operational industrial environments. Moving forward, we aim to scale this platform from bench scale to pilot and industrial scale bioreactors, with projected production reaching up to 42 gram per liter. This clearly positions our innovation as a scalable and industry-ready solution for next-generation biofuel production. These are some of our research achievements. And also, we have both patent filling and copyright, and together with LOI with industry collaborations. These are some of major references used in our study. That comes to the end of our presentation. Thank you so much for your attention.